Hey everyone, this is Pat Lopez again. I'm the science department chair here at NPC, and I teach biology. And I've got with me this afternoon as well, Dr. Susan Hoffman, and I'm particularly thrilled to have her with us because she teaches microbiology here, and her degree is actually in medical microbiology
And immunology.
and immunology. Fantastic, even better. So we're recording this on Sunday, December 20th. And, Susan, when I first started hearing about the possibility of vaccines and this idea of an mRNA vaccine came up, and I looked it up and I thought, wow, this is brilliant. And so probably a lot of people have heard that the first two vaccines to be approved for use in the United States, the Pfizer vaccine and the Moderna vaccine that just got approved Friday night, are a new type of vaccine. They use messenger RNA, or mRNA as we call it. And what's particularly exciting to me is that that's one of the pieces that we teach in our Bio 181 course, right? And so we know how it works in principle. So it was really thrilling to know that that's what's going into these new vaccine types. So,
Yes.
but won't you give us a little bit of background on just sort of the basic biology of what mRNA is and how it works?
Right. And before I do that, I'll give you a little bit of background on vaccines themselves, and just a little bit about the development of vaccine and why the immune system, why it's just vaccines work so well, and why the use of this new vaccine, which is an mRNA vaccine, is just such a great advance in the field of immunology. And so it's very exciting to immunologists that this is being used now. But let me give you guys just a little bit of background on what we're going to be talking about. And everybody out there knows a little bit. They've heard of the immune system, they've heard of DNA, and we're going to be talking about all of that in just a minute. So in the traditionally, vaccines have been made of usually either inactivated or killed microbes, right? And so they can inject them right into the body, and then the immune system will recognize them and they will raise a response. So that once your immune system does recognize it, it will make antibodies, and the cells of the immune system will make antibodies and what are called memory cells. And so if the real microbe comes along, the immune system is already armed and ready to prevent that microbe from causing damage to you. You won't even get infected.
So Susan, quick question on this cool picture here. The little Y-shaped things, those are...
Yeah, so these are antibodies.
Okay.
Antibodies are amazing things. They're little proteins. And what the antibody will do is, for example, this is a COVID virus. And the antibody most commonly will bind to, this is called a spike protein. And this protein will bind to us. And so if the antibody can bind to the spike protein, then it cannot bind to us.
So it just kind of gums up the works.
It gums up the
Okay,
works.
got it.
So antibodies have a lot of function, but the main function that they have is what's called neutralization. So think about it neutralizing your enemy. It just takes it out. It can't work anymore. You're kneecapping the virus. So those are the most common ways that vaccines are made. But another really common way for vaccines to be made is, instead of using the entire virus, either inactivated or other kind of microbe, what they commonly do is just use a part of the microbe itself. And so in this particular case, if we wanted to immunize against COVID, we would use the spike protein that it would use to enter our cells. And so if we made just a whole bunch of spike proteins and then immunize somebody with it, they would raise antibodies to the spike protein. Then when the real virus came along, then we'd have our memory cells and antibodies, and the virus would not be able to get purchase or get a foothold. So that, again, in summary, the way that vaccines have traditionally been made is they'll grow up the virus, they'll inactivate it, and then they'll inject it in somebody. And even though the virus is dead, it raises an immune response so that when the real deal comes along, the immune system has already recognized it as an invader. Or more long lasting on the shelf life, you can just immunize with just a portion of it. And it's great because they last a long time in the doctor's office. The immune system reacts just fine to this. You get really long lasting immunity. You don't have to worry about growing up the whole virus. You can genetically engineer just parts of them. Oftentimes when you grow whole viruses, you need to grow them in, say, a chicken egg or some other kind of animal. And some people have allergies to them. And so these, what are called subunit vaccines, are very good because people don't have very many reactions to them, and they're long-lived and they're stable. There's a big downside, however. They take a long time to develop. They take a long time to get to produce and develop and research, and then getting into production. It could take years for them to get into production. There's a lot that goes into it. What's very exciting about nowadays is that proteins are made by a code of DNA, and the particle that the DNA uses is called RNA. And so what they've done is, instead of actually using a vaccine that's a subunit of the protein, they just use the mRNA itself. It's very elegant.
Cool.
But what the thing is that, so people can really understand this concept that if you immunize somebody with just a particle of the invader, it will make you immune. But people don't really understand, like, that extra step, the messenger RNA part, and they don't really understand why that would work as a vaccine, because maybe they're just a little bit unclear about what messenger RNA is. So the next thing, what I'd like to do is just explore just a little bit about what is messenger RNA. So this is just something that I drew. So we have cells in our body, and they have a cell membrane. And then typically all of the cells in our body have a nucleus with DNA. And everyone knows that we have chromosomes and DNA. And DNA, that's what makes you, you. So it will determine what quality your hair is, your hair color, your eye color. It will determine what kind of enzymes you make in your body. So for example, some people can digest lactose. That means you're lactose intolerant. And so that's all determined by your DNA.
And all organisms have that genetic material.
All life has
Right.
DNA. And so, but your DNA determines what proteins are going to be made that are going to do the function and support of the cell. So the survival of the cell depends on the proteins that are coded for by the DNA. Well, here's the thing. The DNA is in the nucleus. It's huge. It's a huge molecule. It never leaves the nucleus. And proteins are made in the outside of the nucleus, called the cytoplasm. So the proteins are made in this area, yet the DNA stays in the nucleus. Well, the nature solves this problem very elegantly by, instead of having the DNA go out to the cytoplasm, what the DNA will actually do is just make a small copy of itself. And they call this molecule, it's a temporary molecule called RNA. So the DNA, if a protein needs to be made in the cytoplasm of the cell, it will code a small molecule, this messenger RNA, and then that messenger RNA will exit the nucleus, and it will code for the protein to be made from separate amino acids in the cell.
So hence the moniker messenger RNA, right? It carries
Yes,
the message from DNA to the machinery that actually puts together that spike protein.
right. And so what they say, the analogy that they use is that the DNA is like a big library, a library of blueprints that cannot leave that facility, because we don't want to we can't have it damaged or manipulated in any way. So a courier comes to the library, makes a copy of that blueprint, and then goes out to the building site and then builds that protein from that
So
message.
can I just share with you the analogy that I use when I teach this?
Yeah.
Because my sister lives near one of the Ben and Jerry's ice cream factories in Vermont. So I love that. So I sort of use the cell as the Ben and Jerry's factory, ice cream factory. And, I imagine that there's like a book of master recipes for all the flavors, your Cherry Garcia and your Americone Dream and fish food and all of that stuff. And I imagine that, in my mind, that big book is like in a vault in the CEO's
Yes,
office, right?
that's
And
right.
that's like the
It's
nucleus
in the safe.
in the safe, right? But
And it,
like if it's in the safe, the factory workers can't make the ice cream. And plus, they don't make every flavor every day, right? So on the day that they make pistachio flavor, for example, they'll go into the, CEO's office, pull out the big book, make, half dozen copies of pistachio flavor, and then distribute those out onto the factory floor. And then those are like the messenger RNA. So it's just that little piece, meaning, in this case, what I'm using as one ice cream analogy, flavor, or one flavor of ice cream is really directions for making that spike protein, right? So our
exactly.
cells end up making this virus spike protein, which is
Right.
then recognized as foreign by our own immune system.
Right. So instead of our body making the messenger RNA, it's provided to us in the vaccine. So
Super
we're
cool.
kind of we're going around. So we've got all of the code to make us. And so the analogy, the Ben and Jerry's analogy, is they go into the boss's office and they open the safe, and with their phone they take a picture of the recipe, and then they go out and get it on the internet and send it off to California, right? So a copy is made. The original is never messed with.
Right.
And that's the great thing about this vaccine, is because we make messenger RNA ourselves, and we have all of the machinery to make any protein from that messenger RNA. But if we are provided, which is what this vaccine does, if we're provided with the messenger RNA to make the spike protein of the virus, it will produce that spike protein. It will be recognized by the immune system as foreign, and it will raise an immune response. So it'll raise antibodies, and it'll create a memory of that spike protein. The immune system is wonderful. We like to think of it having a brain, but it doesn't. So we say that we've educated. I really like a lot of the terminology about the immune system. If the immune system has not recognized a pathogen before, we say that it's naive.
Right. Right.
And if it has recognized the pathogen, we say that it has been educated.
And these are all biochemical responses.
But these are all biochemical responses.
Super cool.
And so the immune system has recognized the spike protein before. It's raised an immune response. It cannot distinguish whether this was an actual invader or somebody giving it just the spike protein to look at.
Super cool.
So
All right.
that
Well, I'm sure. Go ahead.
so we're out and about, And we don't know if somebody is infected with COVID-19, and we have no clue whether they have it, but our immune system does. Our
Perfect.
immune system does recognize that.
Excellent. Well, I'm sure we'll be talking a lot more about the immune system and more details on this, some of the newer vaccines with things like adjuvants or even if some of the other vaccines that become approved have different mechanisms,
Right.
probably make a video on that as well. So thank you so much, Dr.
Okay,
Hoffman,
you're welcome.
and we'll see you guys
Okay,
next time.
thank you, Professor Lopez.